Today we actually have AblBio's CEO Hoon with us, who has just signed a massive contract. We'll talk further with Dr. Lee.
Thank you.
Well, I don't want to beat around the bush, so here's the big question. How did you first meet Sanofi?
Well, back in 3 years ago, we had initial meeting at the time at the JP Morgan and Bio USA. By that time, we just introduced very early stage of our platform technology and ABL301, the molecule itself. And by that time, we think, that we still have a very early stage molecule, so they show some interest, but was not very serious. And 2 years ago, we also had a contact Sanofi and other big pharma at the JP Morgan and other, the Bio USA and Europe. And they requested they want to have specifically some monkey data. So I think about a year ago, JP Morgan was that time they seriously considered our technology platform as well as the molecule itself. So because I think that we generated and also accumulated a lot of the significant the data which demonstrate the differentiation with other platform technologies such as the BBB penetration platform developed by Genentech and Roche, and the company called Denali Therapeutics in US. I think that we had in our hand some monkey data to demonstrate our platform is a lot better, superior to other the platforms. I think the Sanofi and other big pharma showed the very serious interest a year ago, and then it took the 1 year from the beginning of serious discussion to the end of the sign.
So from initial contact to a couple of weeks ago, that was 2 years?
About 2 years ago, I think, but I think the serious discussion about 1 year.
I'm sure there were some memorable moments making the deal. By memorable, I don't mean anything good. It can be bad. Were there any troubles? How did you overcome it?
I think we had a couple the big pharma which showed the interest our technology, not only Sanofi but also the company I cannot disclose. But I think each company has different style of the evaluation process. So Sanofi especially has a very thorough due diligence. We actually went through first due diligence around the springtime, around the March and April, and I think that we opened the database, and since then they also added additional second due diligence, which means they requested all the study report in English. So I think that some of the study performed at the CRO outside of the research organization, we had the English version report. However, internal study, we have to prepare the very the QA based, in other words, high quality system, make sure that our data generated correctly. So I think that we did a lot of internal the preparation. So that report, second due diligence, was completed around end of July. So I think that it was the very, very difficult, thoroughful the process. And I think around September, October, we initially get the non-binding term sheet. In other words, initially they had some idea how much upfront, how much total package, how much the milestone can be. And I think by that time, not only Sanofi, we had another the partner who we had a continuously discussion. I think we chose the Sanofi main the couple different reasons. I think although it was a very difficult situation, we had a lot of interaction with them because of the the multiple due diligence. So we had a very close interaction with them. And also, the strategically, Sanofi didn't have an alpha-synuclein for targeting Parkinson's disease. So I think that their neurobiology strategy perfectly fit to us. So it was a very, the difficult situation, but strategically perfectly matched. The most difficult, the stage was we called the MTA, Material Transfer Agreement. That time, every single big pharma wants to make sure, bring the molecule internally, and then they want to perform the experiment, whether the experiment ABL did correctly done, make sure our claim, so-called our bispecific antibody, penetrate a lot better than the monoclonal antibody into the brain, and they look at the data, but they want to perform the exactly same experiment on their hand. So we spent about more than 2 months MTA agreement. So in other words, how are we going to do it? The MTA study will be done correctly, or the experiment assay will be done based on our experience, not the Sanofi experience. But at the end, a lot of trial and error. Make sure, the first experiment we succeed, but Sanofi said we got negative. So, then, we discuss again, what happened the experiment, and we realized that there are certain, the discrepancy between our, the assay and their assay. So, a lot of discussion happened. It took, as I said, almost 4.5 months from the beginning of MTA to the end of the completion of the MTA. So I think, at the end, it was a good story. But a lot of tension, a lot of building the trusting. So in other words, I believe the MTA is the building trust. So in other words, I trust you, however, you want to make sure, the experiment was done correctly. So I think it was a long journey, very tough, the period, but worthwhile to go through it, yeah.
I can certainly feel that confidence by the word. Dr. Lee just used the word, we chose Sanofi, and that's a very unique term probably for a lot of Korean companies, because it's a unique concept, choosing a big pharma that would license in.
Yeah, I think, internally we had a confidence on our technology. When we discussed with a lot of big pharma at the JP Morgan and Bio USA, the more we, to show the data, I think they show, a lot of surprising, because at the first, the initial thought from them impression was, I cannot trust your data, because they didn't say we are the Korean biotech, but I think that we had some impression that, we are not the mainstream of the US or Europe biotech or Japanese biotech. So we are Korean biotech. They never heard AblBio. The technology we actually modified and improved a lot of differentiation compared to competitors. So I think that they started believing us. But however, as I said, they need to see by themselves some confidence of a molecule. I think, at the end, we are very, proud of our scientists have spent more than 5 years to build this technology.
It might be a redundant question. AblBio is definitely one of the venture companies known for working in concert with VCs, investors. What is the secret to that?
I think, when we started fundraising and when I consider founding AblBio, I think that I spent a lot of time with venture capital, such as KIP, Korean Investment Partner, and DSC Investment, especially. I think it's not only just money itself. I think I spent a lot of time with them my first brainstorm initial thought, how I'm gonna form AblBio. And then they requested, what is your, the 5 years plan? And, all the other, the short-term and long-term plan. And in not only also, the plan itself, how we actually move forward our technology from the idea. And then what's the end? So I think a lot of, my spending time with venture capital, I see the AblBio can focus on bispecific antibody. And in immuno-oncology program, which we didn't talk about yet, but I think the BBB shuttle, and by that time I had some idea developing anti-synuclein antibody for targeting Parkinson's disease. However, another big hurdle in the brain area is how we actually deliver the molecule, large molecule into the brain. That's why we call the BBB shuttle. So they actually raised the question, how we are actually overcome by your current technology. Then we did a lot of research searching then. Then we realized that Genentech, Roche, and Denali Therapeutic, AstraZeneca, all the big name, had some research already published in the paper. And we actually thought, okay, there are some medical unmet need in the BBB area. So I think that we spent a lot of time there. So I think that was the very good, the interaction with the venture capital with them. And since then, AblBio was able to successful fundraising, Series A, Series B, Series C, and then in the short time, IPO in 34 months in South Korea. But I think, it's a mutual respect. And also, I think, I'm the key driver, AblBio the key driver to develop technology. I do have a lot of venture capital to support and believe in me and AblBio, so that we actually build a lot of trust and then confidence also. And also I think, every year, I can tell, now that every year we demonstrate the progress of our project. In other words, initially 6 years ago, we had only some idea. But we entered the first IND about 3 years ago. And the last year we did the 3 IND into the phase 1 trial. So that was the originally we planned by ourselves, also discussion with venture capital. But AblBio delivered all the promise to the investor, to the market, although it's a still early stage, the candidate. But I think some of the molecule, as I said, we deliver on time, or we deliver earlier than we, the promise. So that way, I think some good, the trust building between investor and AblBio, and also myself. Yeah.
Was it like that the whole time? Like, I see you delivered the plans 100%, but there must be a time where the plan didn't go.
Absolutely,
by plan.
Absolutely. So I think there are a couple projects we already terminated, and I think when we actually first the license out to the Trigger Therapeutic, which now merged with the Compass Therapeutic. The Compass Therapeutic main investor is the ObiMed, the global one of the largest the VC, and I think that molecule. We had a lot of bumpy road. I mean, when we developed, there were definitely competitor called AbbVie and other, the big pharma. Then, the target in the clinic had some, the side effect shown by many different companies already ahead of us. Then I had a lot of skepticism by investor that what's the major differentiation we call ABL001, which is the bispecific antibody targeting VEGF and DLL4, currently in the phase 2. And by the time we designed some, we call affinity modulation that by manipulating certain target with a lower, the affinity, we believe we can reduce the toxicity. And no one believed that at the time. So everybody looking for high affinity molecule. And at the end, in the clinic, the epi, which had a high affinity to DLL4 target, showed a lot of clinical side effect. Whereas ABL001 still show we call the enough therapeutic index, which means the still manageable side effect. There is no any oncology molecule, no side effect. So there are certain manageable side effects. I think there are a lot of try and lesson that we have to spend a lot of time for research and translation research to demonstrate the differentiation. What's the best way we can show the differentiation? And then in the clinic, at the end, the human trial show our molecule seems to behave better than our competitor. So that was the one of the example. We had a lot of the difficult time, and now it moved to phase 2. It's a good time, and also when we developed this grab body BBB penetration. We actually demonstrate, the novel target we call IGF-1R, not transferrin receptor. Transferrin receptor target is the typical BBB shuttle used by most of big pharma. Then when we say, that we have a novel target, and a lot of skepticism too, the show me the data and demonstrate why you believe your target is better than transferrin. So I think, a lot of science. There is a trial and error, skepticism, demonstrate the data, so we can overcome the skepticism. And also demonstrate the differentiation is the key. So a lot of big pharma want to see the differentiation. So I think we are able to do that by having a lot of failure. Yeah.
So I guess the message is by taking a detour, you can actually go faster.
Yeah.
I think that's
or I can say by taking long journey, but at the end, it's the best way to go. So in other words, I think I don't want to take the shortcut. I think there are shortcuts. However, when I look at all the clinical trial, preclinical development, there is no shortcut. In science, we have to go through 1 to 100 steps and demonstrate every single step. Not only research, but also manufacturing part. We have to demonstrate our molecule is stable, our molecule is manufacturable, our molecule and generating CMC is economically valuable. So all the process, in my opinion, you gotta go through it.
Yeah.
There is no
I definitely agree with that.
Yeah.
So this licensing out deal wasn't your first. There was Trigger Therapeutics. When you first display your pipeline, is it always through those conventions like JPM conference, or was there a licensing out deal where you used a commercial law firm or an agency? AblBio, we do have a 2 business development person in San Francisco area, Silicon Valley area. So 2 of them has the MBA degree, business degree from UC Berkeley and Michigan, and I think that they are highly experienced. So we never use any outside of agent. However, I think the way we had a lot of interaction with different company for licensing opportunity, ABLBio was able to generate a very good, the publication. Last 2 years, we've been published every year 5 different journals. Whatever the publishable data, we encourage. I encourage to the scientists, you have to publish. The publication in the conference
and also actual, the journal publication. Really one of the good way big pharma or other company approach to us. And then they said, we look at your abstract and they want to discuss with us. So the reason why I encourage that is back into my industry experience in US, especially Chiron, currently Novartis, and Genentech, both companies, the Silicon Valley area, they are highly dedicated to the research. They emphasize publication. Once you have enough data, you publish. As long as you have enough, the patent protection. So I think and that's the one way big pharma also demonstrate their technology is good. Their molecule is superior to others. So we've been encouraged to publish. So it's been ongoing. So we've been 2022, we're gonna publish another 6 papers. And we may publish maybe 6 or 10 abstract poster in the different conference. So I think that this is another way we have a great interaction and initial contact from big pharma.
That's a good information for the venture companies in Korea. Other than that, it's known that you have worked for several global companies already, pharmaceutical companies. You've just mentioned Chiron in the US, AstraZeneca, Genentech, and Excelsis. Other than what you have mentioned just now, what experiences and know-how have you gained from working for those institutions?
I think, at Chiron, I was the junior scientist. I was the bench working scientist. I do everyday pipetting. At the first year, I don't manage people. I had one technician, which is a senior technician, who helped me a lot. And I think we actually work together, the task, the project team asked to do. And then I and, the Cheryl Goldbank, my senior, the research associate, show a lot of progress, and my big boss, liked our progress. And I was involved in the first small molecule kinase inhibitor, Chiron, push it toward the IND. Chiron is well-known vaccine company, HCV, HBV. Chiron is well-known in the virus area, oncology. The project I was involved was the first IND molecule Chiron developed. So I learned a lot of the pharmacology stuff, and then PK/PD modeling. And my big boss gave me some opportunity called translational research, so that we are able to develop we call the biomarker to support the patient selection for the molecule, and we demonstrated the in vivo study. So it was a good journey for me to learn from the beginning to the IND and phase 1. In 5 years, I learned a lot, and because of that project, Big Boss gave me, okay, this is your second project. That's the antibody project. We call the anti-CD40, which is the immuno-oncology target. By that time, I was very fascinated. Small molecule, we have to work together with the chemist and biology. But antibody, biologists develop the idea, we develop the molecule. We don't need a chemist. So it's a different, the opportunity. So I think the Chiron was the first company which gave me, I believe I owe a lot to Chiron, that I learned a lot, from the beginning of discovery to the phase 1. And that was the perfectly good experience. And then I moved to AstraZeneca. Then by the time AstraZeneca was so, ambitious that they want to develop the 20 different oncology targets by working with a company called Apgenix. Apgenix acquired by Amgen, 10 years ago. And they had some antibody technology. AstraZeneca and Apgenix co-developed certain, the 20 plus target for oncology. So that was the more project management and also more depth understanding of antibody, the development. And that was a good experience too. But, the California, I just like more California than East Coast in terms of culturally also weather-wise. And I moved back to Bay Area to continue working at the Genentech. And that was the another big open eye for me, because I was the group leader of the biomarker, which cover entire Genentech oncology program. So it's not only small molecule, but also the large molecule, and also some ADC. So I was not involved in the early discovery. However, I was involved in the translational research, discover biomarker, and then implement biomarker into the clinical trial, discussion with the clinician, how we actually work together to support the phase 1 trial, something like that. So that broad experience really helped me to understand more, the larger range of the drug development process. And actually, she's a small molecule company also. So I think through all, the 4 different companies, each company has a different taste, different way of the drug development, and different way of the human management. I think based on all different the multiple cultures, I call industry cultures, and it really helped for me to become a leader in South Korea. If I were in US, I could be different, but by coming back to Korea, utilizing all the experience, people expect also, I personally should be to become a leader or thought leader or, the management leader, that, to lead, the company or project, etc. So I think, all the good experience, but yes, I had a very hard training at US. So in other words, I think one of my big boss at Chiron, recently she was the head of R&D at AstraZeneca MedImmune, and Bahija Jaral became a CEO of the Immunocore. And last week they announced FDA approval of the TCR-CD3 bispecific antibody. She actually trained me, working hard, and she just demanding a lot. So, but I think the Bay Area, San Francisco, Silicon Valley culture is, I should call, demanding. More and more. I bring some, they say more. So I think culturally, it really helped me also to lead AblBio, and we should deliver all the things on time.
Now we're more curious about the current AblBio. Could you tell us more about GrabBody B and GrabBody T in detail?
Yeah. I think the GrabBody B is when we work at the outside company to name the GrabBody. So we had multiple choices. However, we just like GrabBody. It's like the graphing. Yeah. So bispecific antibody in GrabBody T is a graphing tumor, and we can engage the T cell. So that's why we named the GrabBody. Then the T means the T cell engager. So immune the system penetration. GrabBody B means from the BBB, blood-brain barrier from the brain. That's why we call the GrabBody B. So I think the to develop to GrabBody B, as I mentioned earlier, emphasize the differentiation, but there are certainly 3 differentiation we generated. First of all, the target. We are not working on transferrin receptor. We decided IGF-1R receptor, insulin growth factor-like receptor. There are a lot of literature suggested the BBB shuttle can be transferrin receptor, IGF-1R, or IR, insulin receptor, or LRP, etc. There are certain, the potential candidate. But we look at all the target validation we call expression level, not only normal tissue, but also, the patient sample, and also where the expression. So we believe IGF-1R is the novel BBB shuttle target. That's the first differentiation. Second differentiation is the modality. In other words, bispecific antibody has so many different formats. initially we've been working, we call 2+2. In other words, alpha-synuclein part in the upper part, the antibody, and then C-terminal antibody, we conjugate the 2 IGF-1R, the single chain FB, and this is called the bivalent. But by the time Genentech, Roche, and Denali published that, there are certain, called the monovalent. In other words, instead of having 2 BBB shuttle, having 1 BBB shuttle, monovalent, show the better BBB penetration. So modality also we, perform all the experiment. We also confirm that monovalent, which is the asymmetric formation, bispecific antibody shuttle, seems to have a better BBB penetration. Third, the differentiation is the Genentech, Roche, and the Denali Therapeutic, other company. Although they developed the bispecific antibody, they have a short half-life than typical conventional monoclonal antibody. So in other words, if you generate the antibody, monoclonal antibody in the monkey, let's say you have an 8 days half-life, and Genentech half-life is the 4 days. So the key question is, to penetrate the brain, your molecule need to survive wandering around in your body. If you have a short half-life, before you get into the brain, your molecule disappear. So you have to have a long half-life, which is not AblBio original theory. It was the theory by Big Pharma, because they already demonstrate. Surprisingly, we are the only one so far. We did the antibody engineering. Therefore, we increased the half-life in the monkey from 8 days to 12 days. And then at the end, we compare Genentech, Denali, and our BBB shuttle, not only PK, we call PD, the biological effect, efficacy, and longer half-life molecule, which is the ABL-bio body B, showed a lot better efficacy. So I think the 3 differentiation key was the main factor we are very proud of. body B, the platform technology. And the body T is basically 4-1BB immuno-oncology. So it's very, the intellectually difficult area, immuno-oncology, but there's a T cell. The major T cell signal is the CD3. And the secondary signal is the 4-1BB, 4-1BB. A lot of companies are working for 4-1BB. The famous, the CAR-T construct. Also, they have a CD3 construct, and then third generation has a 4-1BB signal. So it's a boosting signal, but it provided some we call the memory T cell. So in other words, even though the memory T cell function is let's say you have the tumor, and long time later, the memory T cell remember your the antigen of the tumor, and then ask T cell to invade and contact and kill the tumor. So it's the long-term memory signal is mediated by 4-1BB. So we actually generate the 4-1BB platform we call body B, body T, and I think that we have multiple projects ongoing. ABL503, which is the PDL1 4-1BB. So it's a famous PD1, PDL1. And second molecule, ABL111, 111, is the Claudin 8.52 4-1BB, which is the more pancreatic tumor and gastric tumor. These two projects, ABL503, ABL111, have been developed with my partner called IMAP, which is the Chinese company, but in the Nasdaq listed. So we spent more than 3 hours from the discovery. Right now in the phase 1 trial, it's a cohort 4. So it's moving forward very well. So I think that we demonstrate preclinically very strong, good significant data. And now we are performing clinical trial phase 1. We are hoping we can show major differentiation compared to GenMap and other big pharma 4-1BB programs. So I think that we are hoping to watch and monitor the progress. And ABL501 is another the bispecific antibody targeting PDL1. and then called the LAG3. So people believe the why PD-1, PD-L1 is important because it is a brake signal for the tumor. So the tumor release the brake signal to the T cell, do not attack to me. So therefore, if you break the signal, then the T cell can, attack the tumor. So that's the first break. LAG3 is considered maybe secondary break or tertiary break, which is another important, the immune checkpoint signal. So we combine together PD-L1, LAG3, and we initiate the clinical trial in South Korea last year. So we are able to demonstrate our platform utilizing Grab-body T, and then moving forward 3 different molecules into the clinical trial. That's another major the milestone for AblBio with 95 people, and we have the ABL001, 3 molecules actively ongoing. We have a very interesting molecule working with Hoon Bio. So we sign up the ABL105, which is the Herceptin and 4-1BB conjugate 4-1B, the bispecific antibody. And Korean biotech, we've been working together, up to now, the GLP tox study. Soon, Hoon will do IND filing. I think this molecule also very good, the differentiation in preclinically, superior to, the ongoing, the competitors. I think, that we are very excited about this molecule progress. So all the other, the Grab-body T-based molecule, I think that we have a very, the fruitful, the product candidate. In 2022 from now, we are hoping we can engage, the big pharma to potential license out this, the Grab-body T-based, the platform, as well as the molecule-based.
Other from that, we need to mention what ABL001 is because it has been with ABL's inception the whole time.
Yeah.
So we need to hear more about the progress. It recently received an approval for phase 2 in the US. Could you abbreviate?
I think as I mentioned, this ABL001 is the VEGF and DLL4 bispecific antibody. I think a couple years ago when we did the IND filing in Korea, that was the first bispecific molecule any Korean biotech did the human clinical trial. So that was a pretty good major milestone. It has a lot of history. So after I returned back to Korea, I was the co-founder of the Pharmepsin, which is the co-stock listed. And then about the 4.5 years later, I left Pharmepsin and joined to Hanwha Bio. They had the biosimilar group and novel drug development. The new drug development group was developing this ABL501, we call previously Hanwha HT105. We also published this, the data in the journal called the MAB. And I think, when Hanwha closed, I think this molecule, the transfer to Binex, and then I signed up with Binex and set up the, the CMC contract, and also we can continue develop. I think almost, the people will never see this ABL001, but actually ABLBio revive, the previously, the molecule developed by Hanwha. And we actually IND filing right after we, the founded company, and it was very fortunate because we previously up to development of to the IND ready molecule, we had a Korean government grant called Sanjabu, and after that we had another Korean government called NOV, National Onco Venture. 항암신약사업단, that they supported more than 45, the 4.5, the million US dollar, 45억, 50억, something like that. So I think as a startup, this more than 4.5 million Korean government grant really help us to manufacture GMP and also lead the clinical trial. And also by that time, AblBio didn't have any clinical group at all. So, we as a research scientists work together with NOV and supported by NOV, we are able to finish all the clinical trial phase 1. So, I think it was a good journey between private company, big support from Korean government grant. So it really helped. As I said earlier, during phase 1, we had a lot of skepticism about ABL001, whether do we really have a differentiation? Is it just a one-off day you want to show to the people? Or you truly believe? And I think that AblBio did a lot of translation research. We look for the biomarker. We also, in the patient-derived efficacy model, we demonstrated our molecule seems to behave a lot better than other competitor. So I think that we collected all the data, but right now, after finish all the phase 1B, I think that we have a very strong clinical data suggesting this molecule seems to be a lot better than our competitor. And as you mentioned that our partner, Compass Therapeutic in global, and LP Science in China, and Handok in Korea, all of them entered the phase 2. And especially Compass and Handok announced that they want to focus on more BTC, biliary tract cancer. It can expand to colorectal cancer, it can expand to gastric cancer and ovarian cancer. But I think the biliary tract cancer, BTC, has a lot of medical unmet need area. So no drug available this time. The data Handok generated, also phase 1B ABL generated, and I think the ongoing phase 2, in my opinion, compared to all the different angiogenic inhibitor, I think that we have a pretty much superior efficacy, although we have to still continuously monitor safety. But the general consensus is this molecule seems to behave better than Avastin or better than angiogenic inhibitor. So I think that this is our big hope that in Korea, industry has a lot of struggle at this moment. Not only all the biotech sector, but also whatever any biotech, Korean biotech license out to major big pharma, the company, we didn't really see the progress of molecule moving from phase 1 to phase 2, from phase 2 to phase 3. So currently, Yuhan molecule seems to be great with Janssen. Our molecule is one of the good examples also. We license out, the early stage, and then we demonstrate good phase 1 data. Now our partner, Compass, which has a tremendous of cash generation, the results. So they are able to conduct a different phase 2. So I think this is another big opportunity we like to see whether or not ABL001 can be a good example, another license of their success, but also we can provide some benefit in BTC patient which not any drug is available at this moment. So we are very excited about this, the ABL001 too.
Well, we need to wrap up now, and this will be my last question. Tell me about AblBio's plans going forward. Many presume, especially in the media, that AblBio will keep pursuing this licensing out strategy for the time being.
Yeah.
Did you ever think about research to commercialization? Why are you pursuing license out strategy only right now?
I think there are certainly good example we can find in US biotech as well as the biotech in Europe. So although everybody knows Genentech, Amgen, there are the 30 plus years of history. But ongoing, the very the famous biotech called US-based Regeneron and ADC technology company previously called Sierra Genetics, now Seagen, and European company called Genmab and Genco. All of them are antibody company. or my previous company, the Excelsis is a small molecule-based company. Every single company I described was the platform-based company. They licensed their platform to Big Pharma. Seattle Genetics licensed their ADC technology to Genentech, Pfizer, all the other Big Pharma. Then, certain time, the revenue generation came from milestone. And then, the certain, like Genentech, the TDMO, not Seattle Genetics, but Immunogenic, but this ADC technology, bispecific antibody, monoclonal antibody technology generate the revenue for the company I described, so that each company started transformation. Then they also had an internal program. Once they have, the license out, some cash reserve, and also internal resources, human resources for conducting later stage clinical trial. Then they perform their own commercialization process. So now, the Regeneron took 30 years. So back in 2000, they had only 150 people. Now, I don't know, 5,000. Seattle Genetics, when I was in Genentech, I had, we actually licensed in their CD40 molecule from Seattle Genetics and also ADC technology. I was the due diligence team to be there. They struggle a lot, 100 plus people. But now, they have a tremendous of revenue. And GenMab and Genco is the same way. So the reason I'm saying is the Korea Biotech, not only AblBio, K-Bio, need to go through, I believe, this step. As I mentioned earlier, there's no any shortcut. You gotta go through it. You have to have human power. You have to have money. You have to have resources. You also have a good molecule. So if your molecule is bad, whatever resources, you will fail in the later stage clinical trial. So I think the building Korea K-Bio, in my opinion, is still behind to US, the biotech industry. We have to learn the lesson from them. So my plan is, at the end, AblBio wants to be a pharmaceutical company. We like to see the success of our molecule phase 3 trial. The key question is, should it be now or can it be later? And we do have a certain big plan we call ABLBio 2.0. So last 6 years, we call ABLBio 1.0. So now, we had a big success with Sanofi. It's not the end. It's the beginning of our, the big journey. I like to see more opportunity of the GrabBody be platform-based, the license out deal, because I think, as I mentioned earlier, when we discussed with the ABL 301 deal, Sanofi was not the only company. So you can imagine, either way, they didn't believe or they were kind of hesitating, something like that. That's why we, as I just said, the good partner, I believe, Sanofi. We chose Sanofi, Sanofi also chose us. The reason Sanofi chose us is, just imagine, Big Pharma should evaluate all the BBB technology outside, not only AblBio. They actually look at that. They think AblBio technology is the best. But there was another certain partner, the parallel with Sanofi. We are in the, the discussion table. So, they said they'll regret, but we are working on it. And this JP Morgan was a lot different than a year ago, 2 years ago, JP Morgan, because Sanofi, I was told by many people that Sanofi is a well-known, very notorious due diligence company. So they're a French company, different culture. So as I said, it was the very, very difficult process. However, the other big pharma admit that ABLBio went through Sanofi, then we will skip certain periods. So now we have some shortcut due diligence. We have a shortcut MTA. So believe, we are hoping to deliver to all the investors that we can bring, the second deal and third deal based on great-body B. And I think the great-body T, as I mentioned, we do have, the already clinical trials, the stage molecule. We also believe the platform technology. So we are heavily working on that. So I'm hoping, I can say I will deliver by the time, because even though I know, I cannot say anything is a compliance issue, that, that we are heavily working on that, hopefully to deliver great-body T-based, the license deal too, so that, the AblBio can be positioned in the globally well-known bispecific antibody platform technology. And this year, I mean, as you know that we will get the 75 million upfront, and then short-term milestone from Sanofi about 45 million. Korean with 1,440억, and it's a lot of money. And I think that not only that, we have a cash reserve about 60 million, and plus ABL001 milestone. And then, as you remember, we co-developed ADC molecule with LegoChem, and then LegoChem and ABL license out this molecule to Keystone. So we're gonna have the additional milestone from Keystone this year. So combining Sanofi deal, 001 ADC deal milestone, our cash reserve is more than 200 million. So which is the amazing, the cash reserve. And also, since we did IPO in 2018, ABLBio never did any fundraising. So I think this is another, one of the good example that I just want to demonstrate to Korean biotech, the ecosystem, that we can survive by building licensing deal. People are very skeptical about that. I think the skepticism is one of the way I've been dealing with since I founded AblBio. In other words, when I founded AblBio, I claim I'm gonna do bispecific antibody. Everybody was skeptic. Why you do the bispecific antibody? Why not just combination strategy? So no one believed that. But I mean, we deliver ABL001, we deliver 3 additional oncology program, we deliver great-body B. So I think the bispecific antibody no longer strange terminology. It's a great portfolio. Even Genentech, Roche, and every big pharma, their antibody program, more than 80% are the bispecific antibody. And last week or this week, Roche Genentech got an approval of the bispecific antibody in AMD eye disease and immunocore. I think that we are accumulating more data set. Bispecific antibody is one of the key driver for the next generation the therapy. That's the the skepticism, and 4-1BB is another skepticism people, and then we just deliver it. And BBB is another, because a lot of people said, if you treat the antibody in animal, we show the great efficacy. But at the end, all the the aducanumab, all the others, we don't know why it failed. However, one of the reasons now people believe should be the BBB. So I think we had some the big belief before people start to believe. So I think that's another way. I call AblBio, myself and our scientists are the pioneer of the biotech field in Korea. Then I mean, this is our the future plan. So again, the short term, mid term. Short term definitely license out till. Mid term, we're gonna expand from now. By certain point, we're gonna continue clinical development by ourselves or by co-partner. I think the Sanofi phase 1 trial is one of the way we will learn from big pharma. In the long run, to go back to your question, I also want to be the pharmaceutical company which totally independent commercialization. This can be my plan is the 10 years plan. So ABL 2.0 plan can be part of our transformation.
So I guess the take-home message today is no more skepticism, no more what-ifs, and going around a detour of taking a long journey is maybe the short term. It can be the fastest way.
Yeah.
Thank you for joining us.
Thank you so much,
for the interview. I don't want to call it the interview, I want to call it the insightful lecture today.
Thank you so much. Yeah,
Thank you so
I
much.
appreciate it. Yeah, thank you.
I can't wait till the interview goes up.
Okay.
Thank you.
Bye. Yeah.
Well, that's all the honey squid news we have gathered for you today, and I'll be back soon with new and even sweeter issues related to South Korea's bio industry. Please don't forget to subscribe
And like.
and turn on notifications. Bye.
Bye-bye.